Thank you, Ross. Thank you for the introduction, and thank you for coming to my talk. So today I want to talk about two tools from control theory, which I'm really excited about, and show you how they can be useful in studying systems and synthetic biology. So these two tools are namely the transfer function and the sensitivity function. So the first part of this work, if my slide continues. Okay, nice. So the breakdown of today's talk, so it'll be two parts. The first part will be about the transfer function, and the second part will be about the sensitivity function. So I'm gonna talk about each of these tools in the context of a specific problem, and hopefully that will highlight the utility of it as a general tool. So for part one, I'd like to talk about transfer functions of biochemical network motifs. And for part two, I'd like to talk about sensitivity functions of biochemical feedback loops. Those are a bit of a mouthful, and I hope it will make sense at the end of this talk. So the structure for each part, I'd like to start by motivating a biological question on why we even need a tool like this, and then show how this tool can address this question. And we're going to solve an example together, and hopefully by the end of it, it might give you some ideas or it might entertain you on why I like these things so much. So my slide's not, okay, it's working. Okay, so the first part, transfer function of biochemical network motifs. This is some work I did in my PhD, and it was under Professor Andrew Clayton in the cell biophysics lab at Swinburne in Australia. I was also supervised by Professor Sarah Russell and Professor Federico Frescoli. So the first example, I want to start with some examples to motivate why the transfer function even should even be considered in biology. What use does it have? So there are a type of cell line, if you can see my mouse cursor. So these are PC12 cells, and if you give them epidermal growth factor, they grow. Their population increases. But for some reason, if you give it oscillating on and off growth factor at different frequencies, they don't always grow. They do different things. So on the y-axis, I have the growth rate, and on the x-axis is the frequency of the growth factor. And if you give it really low frequency, like slow on, slow off, then the cells die. But then if you give it a little bit higher, the cells thrive. And then if you go too fast, the cells they become stable, they don't die, but if you go too fast again, they start to die again. So there's a hint of this frequency-dependent behavior in these biological cells. And here's another one. You have yeast, and you give it some osmotic pressure, and it changes their growth rate. So again, on the y-axis, I have some growth rate, and on the x-axis is the frequency of your osmolyte. And you can see that for really low or really high, the yeast grows really fast, but there's a specific frequency in the middle where it just suppresses their growth. So again, there's another hint at this biological frequency dependence. And for the physics inclined, this sort of rings a bell of frequency response functions. So the question we want to ask is, how do cells make different cellular decisions for different input frequencies? And so this picture here, you have an input frequency that changes its frequency. So an input that changes its frequency, and the cell somehow reads it, and it makes decision A, B, and C. And so for this, we need the frequency response function, but it's a bit ambiguous here. Like, what does it even mean to have a frequency response function of a cell? So the frequency response function of what? And here I'm going to say it's the cell signaling network. This is, well, what we think is the decision-making apparatus of the cell. So these things transmit signals from the extracellular to the intracellular environment. And by doing so, it regulates the internal biochemistries of the cell. And that's what we think changes. It helps the cell make different decisions. So if you can see, the signal comes in and the receptor reads it, and it changes, it gets propagated through the cell into the middle, and it tells the cell to do something. That's where it makes the decision. So we want to get the frequency response function of these cell signaling networks, but a problem arises, which is, aren't they really complex? Like, they can get really large and infinitely loopy and long, and it's just a mess. So how do you actually tackle a problem like this? And One way is to break it down into their what you call network motifs, the components that build up the network. So over here, we can sort of see that there are these reoccurring network shapes that make up the whole network. And you can build them up. And if you know the individual parts, perhaps you can get a clue, not exactly what it is, but a clue, a hint at what the whole system does in general. So how do you actually approach getting this frequency response? And on the left, I want to sort of highlight our physical intuition. So you have a pendulum and you know how the pendulum acts in time. And you have a model of the system. It swings back and forth and obeys like Newton's equations. And then from there, from calculus, you can sort of derive an equivalent version of that in the frequency domain. And that is the frequency response of the pendulum. That's what is often done in, I guess, the physics class. So now we're going to do the exact same thing for biology. So you have this network motif, which represents your biochemical system, and you have a model for that based on your dynamical equations, your intuition of what the system does. And when you have those equations, just like the physical system, you're going to convert it using the same laws of calculus and logical reasoning into frequency to get this frequency response of that biochemical motif. So the flowchart of how you do this is like follows. You start off with the system, and then from your laws or experimental data, you have the dynamical equations, how it sort of evolves in time. And from this, you do a Laplace transform. This helps you carry what information you have from the time domain into the frequency picture. And then later, you do some really neat algebra to pull out these two functions called m and phi, and these are your frequency response functions. And what m and phi do is that if you look at this system, right, here's your sort of black box biochemical system, and you pump in some sine waves, and this m and phi sort of stretch and they delay this. So green becomes red, and this is what m and phi do. The m stretches it vertically, and the phi, so it gives it a phase shift. And if we're going to put this together to see what it actually does. So here's the system again on the top with the incoming and outgoing waves. And if you put them together, that m is the amount of amplification, and that phi is the amount of time delay the system has caused onto the signal. So you can imagine if you have a growth factor, and this is the, I guess, the protein you observe, this is the amount of amplification, the protein ratio to the growth factor you put in, and that's the time you have to wait. So this is what we mean by frequency response functions in, I guess, the context of biochemistry and biology. So let's do an example together. So gene X makes proteins Y. So here's your system. Gene X makes some proteins Y, and then that somehow degrades. And the dynamical equation for Y, the population of Y, is some amount of gene X, let's say, and minus some amount of protein Y. Very simple model. So we're going to do the Laplace transform. You usually just plug this into a calculator, and you get this funny looking equation. And from this, if you do some like trigonometric algebra, you can pull out these two functions. M equals 1 on square root of this frequency dependence, and phi equals the arctan of this frequency dependence. So here you can see omega, the frequency, you have functions that depend on frequency. So it tells you something about this biochemical system in relation to the frequency of your input. So now I want to plot these, right? So again, the system is on the left, the picture of that, and on the top plot is M, the modulation, and bottom plot is phi, the phase shift. So you can see that at the start, for low frequency inputs in this, say if you're like you're adding something that makes the gene at really low frequencies, then M is 1. So the input and output are, I guess, not suppressed. It's allowed to pass through, and 0 for phi. So there's no time delay. There's no phase shift. And so for really high frequencies, the system can't keep up. You're stimulating, you're pumping the system so fast that the mechanism that it's operating at can't see what you're doing. And so your signal is diminished. You're losing gain. You're sort of putting more effort than it can yield out. And for phi, it becomes phase shifted. It's no longer 0. It's becoming some non-zero value. So because you're operating so fast, the system has to catch up and there's a time delay on what you observed for Y. So you can imagine that again. So for really low frequencies, if you put in X, your Ys look relatively the same because there's no vertical stretching and there's no time delay. But for really high frequencies, you're getting dampening and you're also getting a phase delay. So the wave sort of changes shape. So again, this could be help you design an experiment. If you make a synthetic circuit that has this architecture, you pump something, you observe something, this sort of gives you a rudimentary estimated guess of what you could expect. So we mentioned the idea of like big networks, and big networks in I guess biochemistry, gene regulatory networks, protein interaction networks, often nicely fall under some very nice categories. So simple regulation, which we've just covered, auto regulation, these are like your very simple feedbacks, cascades, where you have like really long chains of simple regulation, feed forward loops. So when you have like two chains, a cascade chain and another simple regulation chain, and feedback loops. These are what I'll talk about in a bit, and that's what I'm doing in my postdoc. So these are 5 of the many. There are a lot of these categories, and if you've looked at Uri Alon's textbooks, you can get the whole compendium of these. So I've sort of computed, and I'm going to display some results for some of these architectures from Uri Alon. So on the left column is the motif category, and the motif category can take on different forms, if you will. So promotion, inhibition are the 2 types of arrows. So simple regulation can have 2 forms, auto-regulation can have 2 forms, cascades can have 2 forms. And so the transfer functions is I've put on the next columns. And I want to just run through how by increasing the complexity of the network, you sort of get really rich behaviors. So for example, originally you just have your simple regulations, you just have, something low pass filter for M, and then something going from 0 to phase shifting by pi on 2. But then later, when you have auto regulation, which is like one step above complexity of simple regulation, you start to have these, I guess you could call them bifurcations, where depending on your choice of parameters, you can start with a different phase shift value, but the system sort of converges to a single one for high frequency. So it gives you a clue that by changing the architecture of your system, you can design more rich, controllable signal processing synthetic circuits. So here is feed-forward loops. Again, feed-forward loop, you can have 4 different architectures, and these are the transfer functions. And I sort of want to show you again how by changing the shape of the network, you can get these other behaviors. So here we've observed a peak, which wasn't really seen in our example before because it wasn't complex enough. And over here, you see different types of, I guess, bifurcations or initial conditions. And here is the feedback loop. So feedback loop on the left, these are the different types of architectures, and these are the expressions for their transfer functions. And here are, again, the different shapes that come out of just varying the architectures. And so for this part, I want to stress these take-home messages, which are the cell sometimes exhibits phenotypic behaviors that depend on input frequency, and perhaps the signaling pathways are responsible, they can help us see why. So the transfer function is a tool that lets us study this frequency dependence. And in the overwhelming challenge of large and complex pathways, we can study the frequency dependence of their motifs to get a clue on what the system does. There's a lot of, I guess, limitations and implications on this, which I will be happy to discuss later. And this is a completely general method that applies to any dynamical system. I've just chosen the network motif problem as a way to illustrate it, but it works for any dynamical system or any network of interactions, whether that's molecular, cellular, or on the organism level. It's just a dynamical tool. So the second part is what I'm doing with Anya on my postdoc. So this is the sensitivity of biochemical feedback loops. And this was possible thanks to our other students in the lab, Anne and Tasvia, and my PI, Anya. So I'd like to again start off with some examples that motivate the utility and the need for this tool in biochemistry or biology. So here's some gene X, and that makes some set of proteins X. And after a while, these proteins coalesce, they form a cluster, and when they do form a cluster, they are allowed to interact with gene X again. They inhibit it. All while this is happening, the proteins, they degrade, and let's call the collective rate of production and degradation alpha. And so the rates, this is just I guess, the dummy model that we're going to make. The rate of X over time is given by this feedback mechanism and this degradation mechanism. And it's often drawn in textbooks like this. And again, red and green are illustrated accordingly. And this is called negative autoregulation, like the canonical simplest form of a feedback. We've just established autoregulation before. It's like one of the motifs, and it's really important. So again, here's a second example. Gene X makes some protein X, and these again coalesce, and this time they promote gene X. So again, all while this is happening, the collective rate of production and degradation we call alpha, and I guess the time dependence or dynamical equation of X is dependent on this feedback loop and this degradation mechanism. This is drawn as follows. So the average change again. And again, red and green are illustrated accordingly. And this is called positive autoregulation. So with those two established, I want to go to a slightly more complicated example. So this is the same picture we just drew before. This is negative autoregulation. Remember how gene X made some proteins, and then that coalesced, and that sort of inhibited gene X. So now I'm going to introduce a second one. So here's two separate feedback loops, nothing crazy yet. And suppose each of these clusters, the outputs of each separate loop become the inputs of the other, like this. So we've just combined two loops into one, and this collective loop is often what's called a toggle switch. So while all this is happening, let's allow the protein to degrade at some collective rate gamma. And so the equations that describe our populations are as follows. dx dt, describing the population of protein X, is given by this loop and this degradation mechanism. And for Y, it is given by that loop and that degradation mechanism, and accounting for what we've just established as the collective degradation rate. So this is often drawn in textbooks like this, and red and green are illustrated accordingly. And this is called the toggle switch. So throughout all these three examples, this raises a question. So all these systems depend on parameters. And so if I'm going to pull back one of the examples, so here's negative autoregulation with its equation we've just set up. It depends on alpha, which is the ratio of production to degradation, and n, the cooperativity constant. But surely these parameters can vary. They're not the same at all times in all cells. So how do these feedback loops work despite these variations? And now I'm going to try to be funny. So here's the picture of a cell trying to maintain its function while its environment, the changing conditions, are causing it to adapt. And this is like, I guess, the feedback loop. So the cell is the feedback loop, and this treadmill is like all the changing conditions that are forcing it to somehow work. It has to be robust. And that's the key word, robustness or sensitivity. So how sensitive are feedback loops to variations in their biochemical parameters? How much can it adapt? That's the question we want to ask. And we need a metric for this. And this metric is called the sensitivity function. So suppose F represents the concentration of some biochemical species, and that depends on some parameter theta. Let's say how dilute the solution in the cell is, or how quickly it can bind, how strong the affinity is. So then the sensitivity of F due to these parameters are, and I'll call it S for sensitivity, theta of F, equals the derivative of f with respect to theta times theta on f. So it's basically the rate of change normalized, which basically just means it's the relative change of the concentration with ratio to the relative change in the parameter that we're interested in. And for the context of this talk, we're going to look at steady state concentrations. I guess there's a talk to be had about generalizing this to non-steady states. But for now, for the context of this talk, let's look at steady state concentrations. So where does this idea come from? And it comes from this principle that in a system with feedback, it is physically impossible to be robust in all the parameters of your system. And sort of your cell has to assign what to be sensitive in and what to be robust in. And by finding out where the sensitivity is allocated, perhaps it can help us to understand the system design, whether that be by nature or perhaps we want to make a system. So this is from a paper by Kamash, and they look at, this is negative autoregulation. So here's the gene and it makes some protein and that sort of coalesces over here and then it goes back to interact with the gene, what we just talked about before. And here they find that sensitivity is allocated to cooperativity. So the degree to which these proteins coalesce, that's where it's sensitive in. And it's really robust in its diffusion and degradation. So this part here, where it spreads out, that's really robust. So this is insightful because it's harder to manipulate cooperativity if you're doing an experiment or something. Like you can just dilute it and change the diffusion and degradation, but to change how things bind to each other, that's harder. And so you sort of see that the cell has hidden its weakness in the thing it's robust in. So it's harder to access its weakness. So what about for the other examples we've talked about, right? So we talked about positive autoregulation and the feedback, the toggle switch, the double negative. So the job for the rest of this talk is to see how sensitive these feedback loops that we've established before are to variations in their biochemical parameters. And the method we're going to use is this sensitivity function we've just established. So again, just to recap, the 3 case studies are the negative, positive autoregulations and the toggle switch. And I've chosen them because they have 1, 2, and 3 steady states respectively. So we're just going to go from a really easy case to a more hands-on case. So example 1, negative autoregulation. So here is, so we're going to extend Kamash's results and make some more inferences. So here is negative autoregulation with its equations again. And we see that it's dependent on the ratio of production to degradation. We're going to call that the feedback strength for now, and the cooperativity constant. So to see what this system does, just to have an insight, it pushes an initial condition, initial concentration, into some long time steady state. And we can see this steady state if you plot the dynamical equation we have on the left as a function of concentration. So you can see that there's one point where it hits the 0. That's the steady state. And we want to see where this red dot, this point, how sensitive it is to changes in your feedback strength, alpha, and your cooperativity, n. So if you look at this plot, this is how sensitive that red dot from before was to the feedback strength alpha. So on the vertical axis, I have alpha, and on the horizontal axis, I have the cooperativity constant n. And in the middle is a heat map, and the brighter it is, the more sensitive, the darker it is, the more robust. And you can see that robustness in this circuit is allocated for states, or I guess realizations of the circuit where alpha and n are large. So the system has really strong feedback and its cooperativity constant is really large. Whereas if both were small, or either of them were small, then it's really sensitive. That's where the system has allocated its sensitivity. So it's strong here and it's weak there, I guess, as a lack of better words. So here is, we're going to see how sensitive the system is to cooperativity constant n. So that red steady state dot that we saw before. Here is the heat map with alpha on the vertical axis, the feedback strength and cooperativity on the horizontal axis. And we can see that it's darker for high alpha and brighter for low alpha. So it's really robust for strong feedback strength and really sensitive for low feedback strength. And I guess this sort of makes sense. If you're having something cycle on itself and it doesn't cycle strong enough, it's vulnerable to external disturbances. So negative autoregulation, as summary, is robust to feedback strength when cooperativity is high, and it's insensitive to cooperativity when feedback strength is low. So these are the two deductions we've made just from that sensitivity tool. Example 2 is positive autoregulation. So here, again, recall the diagram and the dynamical equation describing the population of X. And it's dependent on the feedback strength and cooperativity N. And again, what it does in time is that it pushes an initial condition into some steady state. But I should have shown the previous two plots next to each other. This one takes longer to do that. So by changing the arrow, you sort of change the timescale in which this happens. But so the important thing is it's non-zero steady states. So you can see this if you plot the differential equation for the system. So if you plot dx dt with respect to the concentration, you can see that there's 2 points where the system is stable. It's steady state, blue and red. And we want to see where and how sensitive these are to the feedback strength alpha and the cooperativity constant N. So the first result is that we're going to look at the sensitivity to the feedback strength alpha, and we're going to show that it's robust to intermediate alpha. So we don't really care about N. So we're going to fix it to any arbitrary value, and we're going to vary alpha to see how the effect of changing alpha changes the sensitivity. I'm going to choose N equals 4. So here is the plot. On the vertical axis is the sensitivity, and on the horizontal axis is the feedback strength. And blue and red are the color-coded for the two steady states we saw before, the blue and red steady states. So for the red steady state, it's really sensitive at small alpha, but it's not sensitive at large alpha. For blue, it is really sensitive for small alpha, and it's also really sensitive for large alpha. So collectively, as a system, if you want it to be robust, you need both of the solutions to be robust. And so they're only really kind of robust in the middle, and therefore it is robust as a system as a whole. This circuit is robust to intermediate alpha, just in the middle. So for positive autoregulation, we're going to now look at how sensitive it is to the cooperativity constant N. And we want to show that it's insensitive to large N. So again, we don't really care about alpha, so we're going to fix it to an arbitrary value, and we're going to change N. So here I'm just going to choose alpha equals 2. So here's the plot again of the sensitivity of the two solutions, our two steady states, and on the x-axis is the cooperativity constant N. So red, the red steady state, you can see that it blows up for small n, so it's really sensitive, but it's not sensitive for large n. For the blue steady state, we can see that it's not sensitive at all. It's slightly increasing sensitivity in the middle, but as a whole, it's not. So collectively, as a system, we need both steady states to be robust, for the system to be robust. And so the system, in this case, is robust for large n. So in summary, positive autoregulation is robust to intermediate feedback strength and insensitive to large cooperativity. So our last example is my favorite, the toggle switch. And now we have two species, so we need two differential equations, which we've derived earlier. And this depends on the feedback strength of X, the feedback strength of Y, the cooperativity constant N, and the degradation rate. And to see what this system does in time, just get a feel for what it is. Here I've plotted the concentration of X and Y, blue for X and orange for Y. And you can see that from an initial condition, they start to compete, and eventually one of them dies out and one of them leaves with all So it's a winner-takes-all system. And the toggle switch has 3 non-zero steady states. So we're going to see where this happens. So if you set each of these differential equations to 0, you can see where they're at rest, and the intersection will show you where the steady states are. So here they are for the red curve. That's where the first equation is 0. And for the blue curve, that's where the blue equation is 0. And they intersect at 3 points, the cyan, blue, and green points. These are our 3 steady states, which we would like to see how sensitive they are to the feedback strength and the cooperativity N. So for reasons, I guess I can elaborate later, you can show that the feedback for X and Y are the same, and gamma doesn't play a role in the sensitivity. So you really only care about one of the feedback strengths and the cooperativity N, hence our 2 case studies. So the first case study is how sensitive is the toggle switch to the feedback strength of X? And we're going to show that it's most sensitive to small feedback strength. So here is the X coordinate. So remember how we had X and Y. So here's the species X. And the sensitivity is on the vertical axis, and the feedback strength is on the horizontal axis. So for small feedback strength, at least one, in this case, 2 of the solutions blow up. They're very sensitive. They only plateau when you get to large feedback strengths. So it shows you that for the X species, it is most sensitive to small feedback strength. And similarly, when you do it for Y, then again, sensitivity on the vertical, feedback strength on the horizontal, they blow up for small feedback strength and plateau for large. So collectively, as the system of the whole, if you're looking at X and Y species, you are going to be most sensitive to changes in feedback strength when it's very small. So I want to do this for N now. So toggle switch, how sensitive is it to changing the cooperativity binding strength N? And we're going to show that it's most robust to intermediate values. So again, here is the X species in the loop. And you can see on the vertical, the sensitivity, and on the horizontal, the cooperativity. The system, 2 of the solutions, 2 of the steady states are very sensitive. They blow up for small n, and one of them blow up for large n. So as a whole of the system, the X species is only kind of robust in the middle. And for Y, the sensitivity in the vertical and the cooperativity in the horizontal, it does the same thing. It blows up for small n, and one of them blows up for large n. So as a whole, for both species, the entire toggle switch, it's most robust for intermediate n. And so in summary, the toggle switch is sensitive to small feedback strengths and robust to intermediate cooperativities. So the take-home messages of this part is that feedback loops are essential to the regulation inside cells, yet this intracellular feedback is implemented with changing biochemistries. And so the sensitivity function can help us to understand the quality of feedback regulation. And this in turn helps us to understand design principles of synthetic circuits. And I guess there's further discussion to be had about sensitivity for the transient. So we talked about the steady state concentrations, but what about the transient parts? How do you define a sensitivity function for something that's changing in time? And what happens when your networks get infinitely large? Can you make some strong statements about the properties of these robustness, or can you define a metric that sort of can live up to very complex systems? So that's for further work and further discussion. But this, I hope, has illustrated the utility of a sensitivity function in studying models, especially when you're trying to design an experiment that you don't want to do yet. So both parts 1 and 2 have papers coming out soon. I don't have a preprint for it yet, but there's a QR code and my email if you would like to get in touch, or I can send you a preprint when it's ready. Yeah, thank you for listening.